Hi, my name is Anna Uruena, and this presentation is to discuss the effects of Brefeldin A with retrieval of KDEL tagged proteins, as well as the explanation for treatments of Botox. So Brefeldin A is a fungal macrocyclic lactone, as well as a potent reversible inhibitor of intracellular vesicle formation and protein trafficking between the ER and the Golgi apparatus. It disrupts the traffic of many proteins, and it's commonly utilized as an inhibitor of protein secretion in cellular assays. In other words, it dissociates co-proteins from Golgi membranes and causes the rapid disassembly of the Golgi complex. It is also an inhibitor to the secretion of cytokines by blocking the trafficking of upstream signaling proteins. Figure 1 is an example of this inhibition of intracellular proteins trafficking. So the KDEL, on the other hand, is a receptor that is an integral protein that carries out the retrieval of escaped ER proteins bearing a C-terminal KDEL sequence. This occurs throughout the retrograde traffic mediated by COPI coded transport carriers. In other words, they retrieve escaped chaperones from the C-Golgi and deliver them from the ER or to the ER through COPI mediated vesicular transport. The activated KDEL receptor regulates bidirectional transport between the ER and Golgi complex and from the Golgi to the secretory pathways. And figure 2 indicates or shows a mechanism composed of the KDEL receptor in which the GQ and GS signaling pathways maintains the homeostasis of membrane transport at the Golgi complex. So each Golgi stack has two faces. We focus on the cis phase because it is oriented towards the ER or endoplasmic reticulum. The Golgi compartment closest to the ER is the cis-Golgi network, also known as CGN. This is where newly synthesized lipids and proteins from the ER arrive in order to fuse with the CGN membranes. Each protein contains a specific tag targeting the protein to a transport vesicle that will carry material from one specific place to another. The disintegration of the Golgi, which is caused by the fungal toxin Brefeldin A, ultimately results with there being no vesicle transportation into the ER. So the figures to the right, figure 12A, just kind of shows you a structure of the Golgi at the cis phase. This is where it would take place. The transition vesicles from the ER fuse with the cis-Golgi network here. And figure 12A, these are both from the textbook, indicates that this is what would happen if the trafficking through the ER took place. So if the Brefeldin A wasn't present. Now moving to the Botox subject. What is Botox? Botox, which is also known as botulinum toxin, is a neurotoxic protein that is produced by the bacterium Clostridium botulinum, a protease that cleaves a snare protein required for this function. This bacterium is present in natural settings, including soil, lakes, forests, and in the intestinal tracts of mammal and fish. This toxin is one of the most potent biological toxins known. It interferes with muscle contraction and can cause paralysis if it's present in large quantities or concentrations. And it also requires proper application. So figure 5 is just an image of a normal ACH release versus the disease model or infection with the neurotoxin with Botox, basically. So the image right underneath it, which the arrow is pointing to, just kind of shows the before and after of the application of Botox in the forehead. So when Botox is used correctly and in small concentrations, it does have several medical and cosmetic benefits, such as to control muscle spasm, to treat crossed eyes, eyelid spasms, excessive sweating, and some bladder disorders. It is also used cosmetically to remove wrinkles caused by muscle concentrations in the skin. Now, to kind of understand what takes place with the application of Botox, since Botox is a neurotoxin, it targets the nervous system, which disrupts the nerve signaling process that stimulates muscle contraction. In order for muscles to contract, nerves release ACH or acetylcholine at the junction where the nerve endings meet the muscle cells. So, what Botox does is that it prevents the release of ACH, stopping or reducing the muscle contraction. And this is what allows the muscles to become less stiff. So yeah, this pretty much wraps up the purpose and the description of how low concentration of Botox can be used as a treatment for wrinkles and muscle spasm. Thank you. Have a great day.